வணக்கம், குட் மார்னிங். என் பெயர் டாக்டர் பிரபாகர். இன்னைக்கு வீடியோல நம்ம 2019 யூனிவர்சிட்டி எக்ஸாம்ல ஷார்ட் ஆன்சரா கேட்கப்பட்ட அபாப்டோசிஸ் என்ற கொஸ்டினை பத்தி டிஸ்கஸ் பண்ண போறோம். அபாப்டோசிஸ் மீன்ஸ் natural cell death or programmed cell death. It is the normal phenomenon, normal physiological phenomenon. So every day millions of cells are producing, equal number of the cells are destroying. That destroying process is called as the apoptosis. Okay. So we will go to the topic. Apoptosis is defined as the natural or programmed cell death under genetic control. In Greek, apoptosis means falling leaves. பழைய இலைகள் உதிர்ற மாதிரி. It is also called cell suicide. The apoptosis is a normal phenomenon. It is essential for the normal development of the body. So it is contrast to the necrosis. Necrosis is the pathological cell death. Apoptosis usually does not produce inflammatory reaction in the neighboring tissue, but in the necrosis, inflammatory reaction seen in the neighboring tissue. The purpose of the apoptosis is remove unwanted cells without causing any stress or damage to the neighboring cells. So it is play important role in the cellular homeostasis. So every day 10 million cells are produced in the human body by mitosis. Equal number of the cell die by apoptosis. So it is maintain the cellular homeostasis, and it is removal of the unwanted cells damaged by the infection like virus or toxin. It is important during the fetal development and adult stage. Examples: a large number of the neurons are produced during the central nervous system. That neurons, more than 50% of the neurons are removed by apoptosis during the formation of the synapse between the neurons. Apoptosis is responsible for the removal of the tissues of web space between the fingers and toes during the developmental stage in fetus, and it is also necessary for the regression and disappearance of the Wolffian and Mullerian duct system during the sex differentiation in fetal life. The cells that loses the contact with the neighboring cells or basal lamina in the epithelial tissues dies by the apoptosis. This is essential for the death of the old enterocytes that enter into the intestinal lumen of the intestinal gland. It is also important role in the cyclical removal of inner layer of the endometrium in the female, resulting in the menstruation. Apoptosis also remove the autoaggressive T cells, prevent the autoimmune tissues. So it has many important physiological roles. Apoptosis is activated by either withdrawal of the positive signals or arrival of the negative signals. The withdrawal of the positive signals are necessary. Positive signals are necessary for the long time survival of the cell. The example for the positive signals are nerve growth factors in the neuron and interleukin 2 for the lymphocytes. So this withdrawal of the positive signals activate the apoptosis, and arrival of the negative signals also stimulate the apoptosis, initiate the apoptosis. Negative signals are produced during various events like cellular stress, viral infections, damaged DNA, chemotherapeutic drugs, X-rays, ultraviolet rays, and death receptor ligands like FOS ligand, tumor necrosis factors. These are the negative signals initiate the apoptosis. Death receptor ligands are the substance which binds with a specific cell membrane receptors that initiate the process of apoptosis. The common death receptors ligands are tumor necrosis factors, TNF-alpha, TNF-beta, and FOS ligand, which binds to the receptor called FOS. Death receptors are the cell membrane receptors which receive the death receptor ligands. Well characterized death receptors are tumor necrosis factor receptor 1 and tumor necrosis factor related apoptosis inducing ligand triad. These receptors are called as the DR4 and DR5. DNF receptor 1 is called as the DR4, triad is called as the DR5. Role of mitochondria in apoptosis. External or internal stimuli initiate the apoptosis by activating proteases called caspases. Caspases are cystine dependent aspartate specific proteases. Normally, caspases are suppressed by inhibitor protein called the apoptosis inhibiting factor. When the cells receives the apoptotic stimulus, mitochondria release two protein materials. First one is called as cytochrome C. Second one is second mitochondria derived activator of caspases, that is called as a SMARC or homologo diabolo. Homologo diabolo or SMARC inactivate the apoptosis inhibiting factor. So that the inhibitor is inhibited, that is disinhibition. So it stimulates the apoptosis. During this process, SMARC, diabolo and apoptosis inhibiting factor aggregate to form the apoptosome. The apoptosome which activate the caspases. Cytochrome C also facilitate the caspase activation that initiate the apoptosis. Apoptotic process. Cells shows sequence of morphological changes during the apoptosis. Activated caspases digest the proteins of cytoskeleton and cell shrinks and become round shape. Because of the cell shrinkage, losses in contact with the neighboring cells or surrounding matrix. Chromatin in the nucleus undergo degradation and condensation. Nuclear membrane become discontinuous. Following degradation of the DNA, nucleus breaks into the many nucleosomal units which are called as the chromatin body. Cell membrane breaks and show bubbled appearance. And the cell breaks into the several fragments containing intracellular material including chromatin bodies, organelles of cells. Such cellular fragments are called vesicles or apoptotic bodies. Apoptotic bodies are engulfed by phagocytes. Okay. Within the normal limits, apoptosis are beneficial for the body. Apoptosis within normal limit is beneficial for the body. Too much or too little apoptosis lead to abnormal conditions. Common abnormalities due to too much apoptosis are ischemia, acquired immunodeficiency syndrome, neurodegenerative disease like Alzheimer's disease, and common abnormalities due to little apoptosis is cancer, autoimmune, lymphoproliferative syndrome. Apoptosis pathway. Stress, p53 protein, DNA damage, chemotherapy initiate the intrinsic pathway of the apoptosis that stimulate the mitochondria to release the cytochrome C that activate the initiator caspases 9, and mitochondria also releases the second mitochondria derived activator of the caspases or diabolo that inhibit the apoptosis inhibiting factor. Normally apoptosis inhibiting factor inhibit the caspases 9, that is removed, that inhibition is removed, disinhibition by the SMARC or diabolo. So it initiated the apoptosis. The caspases 9 activate the caspases 3. This is the intrinsic pathway of the apoptosis. The extrinsic pathway of the apoptosis is due to the death receptor ligand that That green color is the death receptor ligand. Here wrongly mentioned death receptor. Okay. So death receptor ligand. This is ligand. This is a death receptor. This is not the ligand. This is the death receptor. This is a death receptor. This is a death receptor ligand. Death receptor ligand is the FOS ligand. Death receptor is the FOS1 receptor. When the death receptor is bound with the death receptor ligand, that initiate the extrinsic pathway, that initiate the caspases 8. The caspases 8 activate the caspases 3 and produces the apoptosis. Morphological changes occurring during the apoptosis. This is the normal cell, nucleus, mitochondria, endoplasmic reticulum, vesicles. So during the apoptosis, cell size is shrinkages and chromatin is condensed. Chromatin is condensed and membrane is blubbing. Cell membrane is blubbing and the nuclear is collapse and intercellular fragments form the apoptotic body formation, that is phagocytosed by the neutrophil and macrophages that remove the apoptotic lysis apoptotic bodies. Summary of apoptosis. Apoptosis is a programmed cell death. It is a normal physiological phenomenon. It is important for the cellular homeostasis and sex differentiation during the fetal life and the adult stage. The apoptosis occur in the two pathways. Intrinsic pathway is mediated through the mitochondria and extrinsic pathway is due to the death receptor is bind with the death receptor ligands that activate the caspases, finally activate the caspases 3, produces the apoptosis. So apoptosis produces the morphological changes in the cell and removed by the phagocytosis. Within the normal limit, phagocytosis is helpful for the human people. So in the increases apoptosis or decreases apoptosis also produces the disease. Thank you for watching this video. If you like this video, share your friends. Thank you.